Good afternoon. Today again I have brought about a new topic, that is prokaryotic transcription. So this topic is included into the MSc Zoology syllabus of the first semester 62 paper. While in BSc Zoology, it is in the 7th semester paper, it is MSc 13 molecular biology, and it is same for all the state universities of the Bihar. It is the same syllabus. So it is applicable to all the state universities of Bihar, including Bihar University, Darbhanga University, Bhagalpur University, Magad University, Pune University, Ara University, and Munger University. So today I will cover prokaryotic transcriptions. So there is a slide. This is a central dogma. You may have heard what is central dogma and how it is described. So in Mali, central dogma of molecular biology. What is actually the molecular biology? In molecular biology, we study three basic steps, three macromolecules: DNA, RNA, proteins. So the DNA is known to replicate and make its own copy by a process that is known as DNA replication. And the synthesis of RNA from DNA is known as transcription, what you see here. And sometimes in certain bacteria, virus, the RNA is the genetic material and it is reverse transcribed to DNA at the time of infection by means of a special kind of enzyme that is known as reverse transcriptase, and this process is known as reverse transcription. And information is stored into DNA is transferred to the RNA by process of transcription, and this RNA information containing information is translated into protein by process known as translation. RNA can also replicate and make new RNA copy. DNA can also replicate, make new DNA copy, but protein cannot make. So central dogma of molecular biology says DNA makes RNA, RNA makes protein. So now we have a certain questions here. So the DNA, RNA, protein. In the earlier word, who came first? Nucleic acid or protein? Nucleic acids are the DNA and RNA molecules. They store information and pass it to the next generation. While proteins are the chemical catalyst and catalyze the transformation of the different chemical reactions known as enzymes. So who came first? It was DNA, RNA or protein? So it is like the egg came first or hen came first. It is like the simple question. Okay. So if the protein, if nucleic acids were coding instruction for protein, how could protein be generated without nucleic acid? Okay. So the question is that if DNA came first, DNA or RNA came first. Okay. So the DNA and RNA came first. Then how they can be copied without enzyme? Enzyme is required to copy, like the DNA polymerase enzyme. RNA polymerase enzymes are required for the copying of DNA or RNA molecules. And these enzymes are protein. If there is no protein, how DNA and RNA will replicate? Another thing is that if protein came first, then how protein can be synthesized, from where it is getting synthesized, who is giving the information? But in both cases, the nucleic acid carry coding instruction for protein, and protein generation requires presence of nucleic acid. So it was like the dilemma question. So who came first, nucleic acid or protein? So it was Thomas Cech in 1981. He discovered certain RNA molecule who serves both as information storage as well as biological catalyst and can cleave without the help of any enzyme. They are named as ribozyme. So earliest life had RNA first, later on protein came, then DNA came. Hence, earliest world on the earth around 3.5 billion years ago was having all the living beings having the RNA as a genome as well as the enzyme. So earlier word was actually RNA word. So it was Thomas Check who described all about these things. Now ribozyme, they are called biological RNA who can catalyze the reaction, also known as ribozyme. We will know about these ribozymes when we will study the RNA, yeah, alternative splicing or RNA editing. We will see there also certain RNA work as a ribozyme. So the earliest life had RNA first, protein came later, then DNA came. Hence the earliest word on earth was RNA. So the RNA, if you take the RNA, how many different types of RNA are there? So as you see, it is the central dogma of the molecular biology, DNA. So transcription makes RNA. RNA can replicate itself. Who replicate? Some viruses. And this RNA converted to protein. DNA from quite the mechanism of transcription, DNA also make different other kind of RNA. What are these RNA? Messenger RNA, ribosomal RNA, transfer RNA. Okay, so they are common to both prokaryotic animal as well as eukaryotic. Both make mRNA, rRNA, tRNA. While certain mRNAs are only produced in the certain RNAs are produced only in eukaryotic animal. They are named as pre-mRNA, small nuclear RNA, small nuclear RNA, small cytoplasmic mRNA, microRNA, small interfering RNA, PB interacting RNA. They are mostly, except the pre-mRNA, are the regulatory in region and they control the expression of particular gene or protein. So all the test kind of RNA are getting transcribed from the DNA itself present in the genome. So for transcription process, what are the requirements? So transcription requires three major components. DNA template on which the transcription has to occur, that provides the template strand and the raw material substrate needed to build a new RNA molecule. What is that raw materials? It is a ATP, energy molecule is needed. Four different types of the nucleotides, basically ATP, UTP, GTP, and CTP. And magnesium ion or magnesium, they are also needed for the proper working of the synthetic enzymes. The transcription apparatus, the name is given as transcription apparatus. It consists of many other proteins which are necessary to catalyze the synthesis of RNA molecules. So the DNA template, what DNA template is the strand of a double DNA molecule. The one of the strand work as a template strand on which RNA molecule binds and do the synthesis. So if you enlarge this portion, you will see there are two strands, upper strand and lower strand. The RNA can be copied and synthesized only from 5' to 3 direction. So it is known as template strand. And opposite to this strand is known as non-template. It is not used transparent. Okay. So the genes, where are genes present? Genes are present in both strands. So what we will see here that there are two strands here, minus strand, plus strands. Template strand, non-template strand. In one case, the gene A and C, they are getting transcribed from the positive strands, and RNA is getting made. Gene A or gene 2. But for the gene B, transcription is happening from the negative strands. So genes present on the may be present on both strands or only one strand, and their transcription will be in the but in only 5' to 3' direction. If you see in this case, the transcription is in the 5' to 3' direction, and genes are in the both cases. And in this one, the gene transcription is from starts from here again from 5 to 3 direction. So RNA transcribed from only one strand. In most organisms, each gene is transcribed from single DNA strand, and different gene may be transcribed from one or the other of the two DNA strands. And under the the thing is that what I have said there, that the template and non-template, these two strands of the DNA. So one strand is also called as a template strand or non-coding strand, and the opposite strand is called coding strand. Why it is called coding strand? Because the sequence of mRNA is exactly same as the coding strand, except the T in DNA is changed into uracil. Thymine changed into uracil. And this is the RNA sequence of the RNA. They are shown in this set of the 333. They are known as codon, and they, upon translation, they produce particular amino acids, and it makes a polypeptide chain. Suppose that it has a, how many codons are there? 1, 2, 3, 4, 5, 6, 7, 8, and they will make a 8 amino acids. So 27, 30, 24 nucleotides in the RNA codes 8 amino acids. And it will be described what is transcription unit. So a transcription unit is a piece of DNA that encodes an RNA. It can be anything like mRNA, ribosomal RNA, tRNA, anything. So transcription unit is a piece of DNA that encodes an RNA molecule, and it has a sequence necessary for its proper transcription also. Each transcription unit includes a promoter that is shown by yellow color, an RNA coding region, then in the last is a terminate. And the promoter region is the region where, actually the regulatory region where the RNA polymerase binds and transcription starts from a point that is known as transcription start site or TSS, and it ends on the transcription termination site. And so it is RNA transcript 5 prime to 3 prime. So what are the promoters? Promoters in prokaryotic cells. So that is known as a Tata box sequence. It is situated minus 10 nucleotide upstream, minus 10 nucleotide upstream from the transcription state sites. So it is a consensus sequence, not necessarily always with the Tata 80. There may be something different also, like if you see TA, TA, TG, AT, TG, TG, and this is known as Brivnau box in case of the prokaryotes, and Hogness box in case of the eukaryote. There is another regulatory sequence present, shown in the colored region, like the green color, at minus 35 base pair upstream. They also assist in the binding of the RNA polymerases, and these two sequences are important, are present just before the every transcription unit or the proper transcription of the gene. So the RNA polymerase, prokaryotic RNA polymerase. So what we say, it is a prokaryotes have only single RNA polymerase, and which transcribe all kind of RNA, that is tRNA, mRNA, and ribosome. It has a 5 permanent unit that is known as core RNA polymerase. Alpha, alpha prime, alpha 1, alpha 2, beta and beta prime, and omega subunit. They make a core enzyme unit, and when they bind with the sigma factor, it is known as RNA polymerase enzyme, holo enzyme. When it is makes the total unit of the 6 unit holo enzyme, then it binds to the promoter sequence on both regions, minus 30, minus 10 promoter sequence and minus 35 regulatory sequence. And upon binding this one, this RNA polymerase now walks through the DNA strand. So, what are the different steps in the transcription? Promoter binding, transcription initiation, elongation, and termination. So, first step is the binding of the RNA polymerase and sigma factor to the promoter. So, promoter RNA polymerase along with sigma factor both bind to the promoter area. And if you see here, now it is named as the holo enzyme. It binds to the minus 35 as well as minus 30 consensus sequence of the promoter. Once it is bind, it creates the bubble-like structure, breaking up the hydrogen bonds between this, and this bubble-like structure is called, means DNA is unbound. This unbound double-stranded DNA molecule. New nucleotides are added, and the transcription is about to start. So once it is open, the nucleotides are coming, nucleotide phosphates are coming, and these NTPs are added, and transcription starts by the 5 prime. It is shown by like this one. So from the start side, the transcription starts. Once transcription starts for 5 to 10 nucleotides, up to 10 nucleotides, then sigma factor of the RNA polymerase dissociates. It has been shown in here. So once it has started, right, the DNA polymerase moves from the promoter to the other sequence, and the sigma factor is required for the initiation of the RNA transcription process. Then comes the elongation, chain elongation. At the end of initiation, RNA polymerase goes and conformation chain, and thereafter it is no longer able to bind the consensus sequence. The polymerase escapes from the promoter and begin transcribing downstream of the template. The sigma subunit is released after initiation, although RNA polymerase may retain sigma throughout translation synthesis. And RNA polymerase progressively unwinds the DNA and the leading downstream is of the translation. Nucleotide to the RNA molecule according to the sequence of the template and rewind the DNA at the tailing end of the bubble. So in bacterial cell at 37 degrees centigrade, RNA polymerase add about 40 nucleotides per second, while during the replication process, the DNA polymerase adds about 1000 to 2000 base pairs per second. So the rate of RNA synthesis is much lower than the DNA synthesis. And where it is 40 nucleotide per second, it is 1000, means 25 times less efficient is the RNA polymerase while transcribing the particular DNA sequence. So the termination, how the DNA transcription terminates. So there is two kind of termination: row factor independent and row factor dependent. So we will talk first about row factor independent method of termination. So what it is seen that at the termination region of the DNA molecule, there are certain inverted repeat sequence. The row dependent terminator contains an inverted repeat followed by string of approximately 6 adenine nucleotide on the template strands. So once they are transcribed, these areas are transcribed by the RNA polymerase. They make a hairpin-like structure. Because they are complementary sequence, they can bind and they make a hairpin-like structure. And what happens at this hairpin-like structure, RNA polymerase pauses. And this poly U structure, which is transcribed from the poly A of the template strand, this is a very unstable bonding here. So the RNA DNA hybrid is released from here and RNA polymerase is detached from the template strand of the DNA and transcription stops. So the it is the sequence in the RNA causes RNA polymerase to follow the DNA. So DNA has the inverted repeat, but DNA had nothing to do with the transcription termination. It is the sequence of the nucleotide in the RNA which is responsible for the termination. So 50% of the prokaryotic transcription starts by this mechanism. So template has inverted repeats. Upon transcription, they form hairpin loop in the mRNA. And template also has a poly-A sequence after inverted repeat. And hairpin structure will be base pairing between the DNA result in the separation of RNA from DNA. RNA polymerase detached from the template. And in row dependent termination, there is a protein that is known as row protein. So this protein is required. While RNA polymerase is synthesizing the RNA molecule and it is reaching to the termination sequence, they do not have any inverted repeats, but they have a sequence, row identity sequence that is known as RUT sequence. Actually, I have been shown here. It's known as RUT sequence, and the row proteins binds there. So DNA sequences that produce a pause in the transcription. They have a two sequence. Two the two features are there in the DNA template sequence at the termination area of the row dependent termination. There are two DNA sequences. that produce a pause in the transcription. Once the transcription reaches up to here, the translation sequence, RNA polymerase halts, stops here, doesn't move. Then they have another DNA sequence that encode a stage of RNA upstream of the terminator that is divide of any secondary structures. So they do not have any inverted repeats here. It is straight ones. Okay, and raw protein has a feature that it will bind to the only straight RNA area of the one which doesn't have any complementarity in between the chain. And once the raw protein binds, it moves along and it catches the RNA polymerase on the top of this. Once RNA polymerase catches the RNA polymerase, raw protein catches the RNA polymerase, then raw protein has a helicase-like activity and it breaks the bond between the RNA and DNA. RNA molecule and DNA is template and it releases the transcript from the transcription bubble. So it is this one. Now I have written another thing that polycystonic mRNA. So in bacteria, a group of genes is often transcribed by a single RNA molecule, which is termed as a polycystonic mRNA. Means a single promoter transcribe more than one single gene. Like the lycopyran, you must have heard about this. They have a 3J, ZYA. Okay, and there are tryptophan also. They have more than two genes in continuation. So same promoter transcribes all the genes in a tandem. So there is only one promoter, and they are known as polycystin. Okay, so summarize the talk. Synthesis of RNA from DNA is called transcription. All kind of RNA are synthesized from DNA. RNA was the first molecule that could also store information and catalyze reactions. In prokaryote, transcriptions occur in the cytoplasm. Transcriptions and translation co-proceed. They occur together. Single RNA polymerase enzyme transcribe all kind of RNA in the prokaryotes. RNA polymerase enzyme is a multimer protein made of 5 core subunits and signal subunit is needed for the initiation, and raw protein is needed for the termination in 50% of the case. The transcription of gene starts after the binding of RNA polymerase enzyme to the promoter sequence. The steps involved in the transcriptions are promoter binding, chain initiation, chain elongation, chain termination, which can be of two types, row dependent and row independent. So transcription could be inhibited by some of the antibiotics. So I sorry for this. So transcription could also be inhibited by certain antibiotics such as rifampicin. I couldn't write it here. So an actinomycin D antibiotic. Rifampicin is known to bind to the RNA polymerase and stop it from sliding through the DNA. While actinomycin D is an antibiotic which intercalates within the DNA double strand and doesn't allow the RNA polymerase to bind to the promoter sequence. So by this, I come to the end of the talk. Thank you everyone. And in my next talk, I will talk about the eukaryotic transcription, post-transcriptional processing, alternative splicing, RNA editing. Thank you.